Hello, everyone, and welcome back to Breakdown of Science. I'm Jan.
I'm Michelle.
And I'm Maruf. Today we're diving into something that honestly sounds like it's from a sci-fi movie: 3D printing blood vessels to help grow artificial organs.
Yep. So, and it's quite cool. So, a research team in Stanford, I believe, was figured out a way to design and 3D print what they call synthetic vascular networks, which are basically just man made systems of tiny blood vessels created with technology. So, the 3D print instead of growing them naturally in a body. So basically, blood vessel systems that can be shaped to fit whatever organ you need: heart, kidney, you name it. And they can more or less custom build these blood vessel trees, structure wise, to match that body system.
Exactly. And the tech they're using is all based on computational fluid dynamics, in short, CFD. So you guys can think of it like a weather forecasting software, but for predicting how liquids such as blood flow through these complex shapes. And it's also based on some pretty advanced optimization software. And it's basically like the best pad GPS for weather design. So they test routes for tiny tubes and then pick the one that delivers the oxygen most efficiently. But the gist of it is that the program kind of grows the vessels inside a 3D model of the organ. It so that it makes sure that every part gets the oxygen it needs. So it's like digital biology meets engineering. You can think of digital biology as like modeling and simulating living systems on a computer before doing them in real life.
And what is the best part of it? The best part of is it's very fast, like ridiculously fast. Like older versions of this kind of modeling would take weeks or even months to finish just one organ, where but this new system is like 230 times faster thanks to better code and smarter ways of handling the 3D shapes. They can now model a full heart with over a million vessels branches in just 5 hours, so to say.
Yeah, 5 hours, it's incredibly fast. I mean, if you've ever seen how dense blood vessel networks are, it's uh, it's something like it's just like something in a human heart, for example. It's a complete maze of tiny tubes, and now they can design that entire maze uh, before needing to do anything else. Uh, so in such a fast amount of time, it's crazy, and it can be used in so many situations that we will go into much later on.
And that's a total game changer. I mean, it means that researchers can try out, different designs, run tests, and make these adjustments way faster than before. So there's no more waiting around for weeks just to see if one version works. And that's totally cool.
Yeah, exactly. It's a huge time saver, So when we are dealing with the complexity of biological systems, every single hour we're saving a massive time, and it's a massive advantage too. And when we like you are working with stuff as complex as organs, that kind of speed makes a massive difference, and it allows for iterative design where you can quickly refine your models based on the simulation and the outcomes of the simulations, much like an engineer like optimizing bridge design for an example.
Exactly. And the coolest part is that they don't just keep it on the computer, they actually 3D printed some of these networks using bioprinters. You can imagine as a 3D printer that instead of using plastic, they print layers of living cells and gels, kind of like icing a cake, but with cells. And these printers, they use bio-ink, which is basically a gel mixed with living cells that can be squeezed through a printer nozzle to build these tissue structures.
Yeah, in one test, they printed out a network with 500 different branches inside a hydrogel. Hydrogel is a water-filled jelly like gel O that holds its shapes and supports printed cells. Rings and it rings with human kidney cells plus 25 teeny tiny vessels running through it, and this basically demonstrates the practical application of their design. It's not just like theoretical, they can actually fabricate these
Mm.
structures.
Yeah. And they then pumped some nutrient-rich fluid. So basically, like a fertilizer run through the pipes, which carried food and oxygen to cells inside of water and minerals to roots. So through these little vessels, basically was a stand-in for blood to see if it would actually keep the nearby cells alive, and surprisingly, it did do that. So the cells closest to the vessel stayed healthy, which is a great sign that these printed networks can actually do their jobs.
And that's definitely, a really good discovery because just printing the shape of an organ isn't enough. And if these cells inside aren't getting oxygen nutrients, the old thing is kind of like pointless. So you need that blood to flow.
So exactly, yeah. These early tests show that the vessels are not just for show; they actually work well enough to support real tissue, at least on a small scale.
We aren't like printing a whole working organs just yet, but this does show that we are getting closer, and the fact that these printed vessels can keep cells alive is a big step in the right direction, because it means that uh, like now we usually just rely on uh, organ donation from people. Sometimes when it comes to like kidneys or parts of livers, the person can still be alive, but in most cases you tragically have to want a person to be dead to be able to harvest their organs for this type of stuff. But with this happening, if this develops as it is currently, uh, it will help, because then you can just go to the part of the hospital, print out the organ in maybe 5 hours, uh, and you just have it ready. And you can also code it so that it matches the recipient's uh, like signature, so that its body's immune system doesn't fight it. And again, it's and that's like a big there's a big challenge for this. It's the But there is a big challenge, and it's vascularization, which is the process of creating or growing blood vessel networks inside of tissues to keep cells alive. So you can't just print solid chunks of tissue. Cells aren't Because cells aren't like little islands. They rely on constant supply of oxygen and nutrients to survive, and that has to be delivered through the blood vessels. And without that network, the inner parts of a printed organ would just die off. So the fact that this current system is keeping that, is being allowed to distribute nutrients, is very incredible.
Exactly. So without these networks, like kind of like building a city without the roads or plumbing. So it definitely looked good on the outside, but nothing would function. So that's why the cells need access to tiny capillaries to exchange, the gases to get the nutrients and remove waste. So, by the way, are like the smallest blood vessels. So they are so thin that the red blood cells can pass through to deliver oxygen. Yeah.
Yeah, and in their models, they managed to keep the spacing that we're talking about incredibly tight. Every cell was within 100 to 150 micrometers of a blood vessel. That's really, really close. So, for context, most human cells start dying if they are more than about 200 micrometers away from a capillary, because diffusions, like a drop of food coloring, we can say, like spreading through water, or whenever the smoke is in the air and it's dispersing around the environment. This diffusion alone cannot keep up with their oxygen needs, so that's why they die. And this precise control over vessel spacing is, we can say, a direct result of their advanced computational modeling, and that ensures optimal nutrient delivery at very cellular level.
Yeah. And when we say tight spacing, we're talking about biological levels of density. So the human heart, or for example, has around 2,500 capillaries per cubic millimeter, and that's like packing thousands of ultra tiny tubes into a very like small sugar cube sized chunk of a tissue. So yeah, if you want to keep the cells alive and functioning, you need that kind of insane vessel density.
Yeah, and at this point, it's not just about survival. With proper vascularization, it's also key to make sure that the different parts of the tissue develop correctly. So you need to make sure that the blood vessels help the signal to cells and guide tissue organization, and even influence how the stem cells turn into the right types of cells.
Yeah, it's one of the reasons organ printing has been so tough up until now. You can print muscle cells or liver cells, sure, but unless you can build a vascular system alongside them, you are left with something that cannot go beyond a certain complexity and size. This breakthrough directly addresses that bottleneck where we had problem before, and it offers viable paths to large, more complex tissue constructs.
Yeah. So this vascular network modeling is kind of like the missing puzzle piece. So it brings us closer to the printing, not just tissues, but also like actual functional life sustaining organs.
Another cool part, they made their code open source, so a model where anyone can just view, use, and contribute to software code, and it's like a community. So we can even access them and can understand if we are interested in how they actually did all of this. And it's now part of the sim vascular platform, and it's, as said, an open source tool researchers use to design and analyze blood vessel networks. So other teams can build on their network too. This open source approach, this definitely fosters collaborations, and it also accelerates different scientific progress by allowing researchers from anywhere in the world to leverage and improve upon their work, whatever they will work on.
Yeah, and speaking of open source, when this it allows it that does allow for the public to be able to access it, and when this study was covered in the news, or more specifically in a recent post from Stanford themselves, in their blog, they did a pretty good job at explaining it. I mean, of course, it's their labs that made it, but they went into good detail about it, because usually when it comes to science communication, things can get a little fuzzy. That's why we have created this podcast, for example, to help with that. But this time in the article that we read from them, they stuck to the main points and got them mostly right. Of course, factually right. They got it because it is what they did. They talked about the crazy speed of the algorithm, the number of the branches they were able to print, and the fact that the cells near these printed vessels stayed alive. And that is a quite a good win towards when it comes to science communication. It's something that has to be more fostered in the science community in general, in publications, and not in just in small update posts and blog posts by the institutes or the universities themselves where they explain it, but just in general in most scientific databases. So it's a very
Yeah,
important win.
I completely agree with you that. What I like the most about is that they didn't just hype it up. They gave actual numbers like modeling a full heart in just 5 hours, which is very incredible, and then printing that 500 branch vascular network that held up
Mm-hmm.
in bio. I mean, it's pretty crazy. I mean, we wanted like for our project, we thought of like printing that what was it called again, plate shaker, and
Yeah,
we
for one of our like
and it was
class
like
projects, it
yeah,
was that we wanted to 3D print
print something.
a
Yeah,
machine for one of our experiments. But when we like, and this is just talking about like plastic, it
yeah,
took
yeah.
how many hours?
I think
Was it?
15 or
Yeah.
more than that. Definitely
I mean,
way longer. So that's this. So like doing a heart, like a full heart in 5 hours is
I
very
mean, they're
crazy.
not again. They're not doing it full hour, but in terms of density, they're more or less doing it. And
Yeah, I mean,
they
it's pretty
probably
insane.
they probably also not just had a very good software for this, but also had very high end machines that worked
Yeah,
in a very uh, like detailed manner and were able to be fast and precise.
yeah.
Well, the 3D printers that we tried to use were the publicly
From
available ones in the library. So,
exactly. Yeah,
but
yeah.
yeah,
But I still think it's really incredible. Yeah. Yeah, and they also, like coming back to the topic, they also even touched on some of the limitations, which
mm-hmm.
is quite fair. For example, they explained that these aren't fully functional vessels yet, as you mentioned before, and right now the printers can only produce basic channel structures, kind of like the skeleton of a vessel, without the muscular walls or the endothelial lining. Um, that the real blood vessels have. By the way, the endothelial lining, it's kind of like a single cell thick skin that coats the inside of real blood vessels, and they control what passes in and out. Yeah.
Yeah, and you're completely right. And to be honest, for a general audience, they walked the line pretty well. So they threw around some technical terms, vascularization, perfusions. So perfusion is the act of pumping fluid through vessels to bathe the tissues in oxygen and nutrients. Bioprinting, but didn't overwhelm people in a broader level, as bioprinting is, I think, well known. They avoided also diving into the heavy fluid dynamics, like the equations and behind the modeling algorithms, which, to be honest, most readers probably don't want or need. But the explanations were still very accurate and informative. This balance is crucial. It informs like overall without oversimplifying and excites without creating
Mm-hmm.
very unrealistic, how do you say, expectations also.
Yeah.
Yeah, and it's definitely really nice to see, science communication doing what it's supposed to do, like getting the core ideas across without distorting the science as we see it most of the times. So yeah, there's definitely excitement, but not misinformation as you see it in general society.
Mm-hmm.
Yeah,
Exactly, it was also perfectly balanced. It was
yeah,
not very complicated. It is definitely a complicated process.
yeah,
They are
definitely.
printing but how they presented it is really very well.
Yeah.
Yeah,
And yeah, again, as you said, honestly, for the kind of complex research that this is, the again, as you said, the public communication was pretty balanced, and the research itself is a massive step forward as well. If we can crack this vascularization problem, like
mm,
if we can figure out how to reliably build these living blood vessel networks, we could be looking at a real breakthrough in organ engineering. Uh, especially if we are able to do these sort of like with the endothelial lining, as you mentioned. If we are able to put these into these structures, then we can start to actually uh, probably use these in surgery or something.
Speaking of surgery, I think this will also really help like to grow transplant-ready organs in a lab. So there will be no more waiting lists, no more rejections from mismatched donors, and also so like the possibilities are like endless and huge.
Mm.
So we're still early, obviously. I mean, these printed vessels don't yet fully mimic the complex structure and the function of natural vasculature, but this work definitely moves us in the right direction, I would say.
Yeah, and it's a very big deal because it can be used as a bridge between several fields, and we have got computational modeling generating biologically relevant designs, fluid dynamics that ensures those vessels function like real ones, and bioprinting turning that into something physical. That kind of interdisciplinary approach is what is going to push biomedical science forward too, and it highlights the power of combining different scientific disciplines to solve incredibly complex, like this example, like this type of biological challenges that we face, want to solve. So yeah.
Mm. Yeah, exactly. And one thing that this sort of idea of bioprinting brings up is like the idea of like personalized organs. Like we are already having this sort of conversation when we're talking about gene editing or genetic modifications, which we'll cover in a future episode, maybe quite soon. Who knows? Maybe follow to stay in the loop. But again, going back, personalized organs. When you have a donor, you can tweak it so that it matches almost perfectly with your organs, with your genetic information, or you can change it so that it runs more efficiently, or make it make your heart a bit bigger, make your lungs a bit bigger to give you more oxygen or whatever. Again, this is still very early on. It's still stuff that has to be worked through, and it would revolutionize medicine, of course. But there will also probably be a commercial side of it, of just people wanting to have the newest and latest organ or whatever. And in one end, it is quite an amazing thing, an interesting idea, but on the other, it is something that's a bit risky. But one thing that is very promising with this is that if these work so that they can be designed specifically for the patient's body, uh, you no longer need immunosuppressants, so mainly drugs that suppress the immune system so that it doesn't attack the newly transplanted organs. And there would be also no donor shortages, and just these custom grown organs when you need them, probably within a few hours, given that currently we have dropped it down from a few months to a few hours already with just the blood vessels.
Yeah, and obviously we have to, stay realistic. I mean, there's definitely a long way to go, but scaling this up, like getting the right tissue types, entering long-term function, blah, 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 it's definitely not going to happen overnight, but it's
Mm-hmm.
definitely exciting to know that we're heading that way. Yeah.
It is. It's absolutely is. It's quite something to. Like, this has been something that, as bioprinting itself, is not something that's new. It's been something that's been researched for a long time, but just now uh, technology is starting to like catch up, both in terms of computational work and also machinery wise, to be able to support this level of precision and speed that are needed. But unfortunately, that is all we have for today's episode. I do hope that you listeners have enjoyed this dive into 3D printed vascular networks, this bioprinting. And please do share this episode with friends, family, that might be interested. Someone that works in cardiovascular surgery, maybe, or cardiology, they probably would be interested in this.
Yeah, and if you got questions or any feedback, please drop them in the comments or send us a message on Instagram. We'd love to hear your thoughts. And yeah, don't forget to follow us on Spotify, YouTube, and Instagram to stay updated for when the next episode is out.
Yeah. Bye.
So,
Bye.
bye.